at populations, how we might try and measure them. We're going to try and measure the distribution and abundance of organisms. We're going to look at techniques to try and do that. Now, where do we start with one of these? Well, there's an old favourite done by a lot of biologists, which is to use a quadrat. Simply put, a quadrat is a square. That's all it is. It's a simple square. We can, in simple versions, which are perhaps 50 centimetres by 50 centimetres, we can put some extra strings across and we can divide these things up. There are in fact better ways of trying to do this, and what I use is a 1 metre quadrat. Now, it's in theory harder to try and look at that using a 1 metre quadrat, but my quadrats are a little bit different than most. So many of the ones that you would see in schools are simply squares, and the simplest ones I've seen are really just squares put together. Mine's a little bit different, so let's see if I can build mine and we'll have a look and see what it is and how it might work, and then I'll show you some samples I've done with it. So let's have a look. Right, okay, so here I am, and what I'm going to try and do here is zoom in a little bit, first of all to show you the top part of my quadrat. Now, my quadrat comes in basically 2 parts, and this is the top part. And on the top part here, what I've got is a simple basically connector, and I can connect either something like a GoPro, or I could put a mobile phone. In fact, what I'm going to use is I'm going to use a small camera. Now, the reason I'm going to use this one rather than anything else is simply the fact that this camera is waterproof, and that seems to have an awful lot of advantages when you're out doing biology. And what I'm going to do is I'm going to position this camera so it's roughly in the center of this frame. Okay, so having got that, can I now see if we can get the rest of the quadrat set up? And what we're going to do is, here I'm going to try and set up the rest of the quadrat, and I'll reduce myself to my knees, and here I've got the quadrat, and it's got 4 sides, and what I can do is put it around the right way, set this one up, then I've got the other side, and I can build this. Perhaps in here wasn't the best place to try and build it for the amount of room that I've got, but it's easier than taking it outside. Trying to do these broadcasts in here is bad enough, but trying to do them streaming live outside proves to be quite difficult. So I've got this quadrat here, and then it's got these 4 arms that come up, and nearly got it there. And now what I can do is let's place this top bit on here. And I'll position the camera back so it's in the right place. Right, so this is not what I wanted to do. Let me just try putting it there. More haste, less speed. Fortunately, this is a very tough camera. It's not going to hold. That's why I put it on first, so it would work nicely. There we are. It's gone on properly this time. And there I've got the camera firmly mounted. And then all I need to do now is to pick up the whole Quadrat, and it will move as one if it doesn't catch on the wires. So I've got a Quadrat. I can now move this, and all I need with this is an extra meter rule so we can see this in action. Well, what we're going to do with this Quadrat is I'm going to try and show you what I've already done. That you would see with it. So we position this so we can take a picture, and then we can take it so we can get the next picture and the next picture. Now by doing this, what we can do is we can get, right, we're just moving the Quadrat. What we can do is we can take a series of pictures rather than just going and trying to count up how many of these different plants in this particular piece of grass. What we can do is we can basically take a picture, and then we can use software here. We can mark the pictures, we can go in and put in how many of each different thing. We can basically record this, and we can use this process and compare something for a number of years. We also get some more interesting pictures as we're going along, and if you're not careful, we can get pictures of the student doing the work as well. So this is this type of Quadrat. So if we then have moved that out the way, now let's have a look at this sort of Quadrat. So what we've got here for this Quadrat, then simply a set to put it up the top, and by positioning the camera here we can take a picture with our Quadrat, and then we simply move the Quadrat either along in a transect, recording what we're doing, or we can do a better way of perhaps of measuring a field by going to have a look and see what we've got in a different way. So let's imagine that we've got a field, and for argument's sake here I'm going to make my field square. What we can do is we can basically divide up this field into a series of 1 meter squares. Once we've got our field drawn here, this is a model of the field, we can then number it, etc. And now I would use something like a good random number generator, and that is going to come up with certain places that we're going to do and use our Quadrat. Then simply with a long measuring tape we can then measure out from this point so far along and so far down, and I can position my Quadrat at that position. What can help a lot nowadays is simply using a GPS. When I started doing biology, we used the very simple sort of Quadrat. We put this one down, we walked around and we tried to guesstimate where we placed it. We did sort of measure things as best we could. But nowadays, by using a camera on one of these and taking random number generators, we can position the GPS here, and that works nicely with this camera, because this camera has the advantage that, as well as being waterproof, it's got a GPS on it, and so I can record exactly where I've taken each of these photographs, and the GPS accurate to about 2 centimeters means that we can be fairly precise where we positioned this in the field, and so we can get a good idea about what's going on. So having got this basic idea that we can now go along and measure different squares on the field, record those, we can work out then how many we've actually taken, and we can try and work out the abundance. So let's have a look and see how we go about doing that. So we want to try and work out here an estimated population. To work out the estimated population, we need to know the number in the sample, and that is going to mean here all of those numbers added together over here the area of the sample. So if a sample was taken perhaps using 5 quadrats with an area here of 5 square meters, and we look at the number in the sample, and let's say I'm looking at, for instance, a picture like this where we're trying to count up the number of yarrow plants in here, then I can count up the number of yarrow plants in my sample, and perhaps that's going to be 40. And so what we're going to then get is estimated number in the population is going to be 8 per square meter. So that's giving me an average here. So we can try and work out the numbers of plants. Now sometimes this doesn't work really well. This type of Quadrat works well for the sort of thing that I'm looking at here. It actually works all right if you're underwater as well, trying to do some of this, but it doesn't really work well for a lot of situations. And so for some we need to use a different sort of Quadrat, and here I've got a photograph of a point Quadrat. Simply, this is a rod normally about a metre in length, and it stands above the ground, and it's got some metal needles, and we drop these metal needles down, and where they fall on what plants, we can decide basically what we've got. So we can look at this sort of idea of placing this down. So let's have a look and see how this one works. So what I have is my stand, and then what we've got is this ability just to drop a needle down, and these are fairly long sort of needles, and sometimes they go through 1 bar, sometimes we put them through 2 bars, and basically we're looking at on the ground what plants we're actually gonna sort of hit this one on. So we might have some plants, and it depends what it touches, and if one of these things happens to miss, then you don't record it. Only if you get sort of a proper hit do you actually record what's going on with this point Quadrat. What's the difference in trying to use them? Well, the point Quadrat here we've got basically is just a way of pointing along a particular line, whereas the other Quadrat, the frame Quadrat, which is then divided up, or doesn't need to be if you've got the camera one, then you can sort of use that one just to try and measure that one in sort of the area per square metre. The advantage of taking photographs of this, of course, is that it's going to record what's in the space, and you can keep it permanently. I find this is useful especially in classes where I might want to try and do something, I'll put a light on, where all the class get the same data. Now the advantage of trying to do this is that it means that students can have a go, record the data, and then we can compare to see if everyone got the same amount of data. And it's amazing how different the data usually is. I give 10 students the same data and I will get 10 different sets. They have to decide certain things, so let's have a look at the sort of things that cause problems with using quadrats. So let's have my quadrat, and we're going to pretend this is 1 meter. Now I've got a plant and I've got another plant. Now, do I count this plant? Do I count that plant? Well, we try and look at the plant to see whether the centre of the plant is within the quadrat, and if it is, I count it. But then we've got another problem, that if I use this quadrat and we move along another meter, I've got to also decide, do I count in this plant there or there? So if we take a stem rule, where the stem is, if it's inside the quadrat, then this one we would count, this one we would discount, and then we would count it when we get into the next one. So we can have that. How about a plant that falls directly on there? Well, we make a decision, and I normally make a decision not to count that one if it falls on the line, usually because that means that when the quadrat frame goes on it, we can't see it very well, and so it's best to discount it. Whereas if it is slightly inside, then I will count it. We get the other problem when you've got perhaps several plants sort of overlapping. It is, can you actually determine how many plants are there? And sometimes it is possible to do that on the ground, whereas with a photograph it's going to be more difficult to actually try and see that. And that certainly doesn't make life easy when you're trying to do this. So if I have a look at perhaps this view, then where I've got a problem down here could be with this plant. If I'm looking here at perhaps dandelions, is this one or is this several? Now, dandelions aren't that difficult, but sometimes the yarrow or some other plants are going to be more difficult to try and determine what they are, and trying to get some perhaps round here much more difficult to determine. But if everyone's got the same rules that they're trying to use and they've all got the same data, then they should get the same answers. And occasionally it does happen. So we've got this basic rule here for using a quadrat. Now, when we're trying to use this, sometimes we can sort of, we've got this picture recorded anyway, so we've got that to record and work out what's happening, and we can refer to that one. If you're doing this by hand, then sometimes drawing these out and trying to position approximately where a plant is can work, and that works especially if you've got a quadrat and this quadrat you can simply then have divided up with perhaps some string across like this. Mine aren't very even, that doesn't really matter. And you could then say, well, yes, I've got one in there, we've got three in there, and I've got two in there and one in there. And you can record each one of these as accurately as you can. So with my camera one, I've got for each one my GPS coordinates. If you've got this quadrat, then we've got some sort of reference frame. You may know that that one is number 1, that one's going to be number 2, number 3, and so on. And then you would record on here that this is sort of frame number 1. Trying to get all this data, trying to store all this data is important when you want to come back and look at what we've got. So that's looking at some sort of plants. We can use other systems with plants which can be basically quite effective. What we've got are other means of trying to record data, and as well as using things like quadrats, we can also use other sorts of means of trying to calculate what we're looking at. If you're in perhaps a wood, counting up the number of trees, it's not going to work well with a quadrat, but it can work with a GPS, assuming you can get a signal, or you can try other means of sort of measuring things. And there are a whole variety now of bits of equipment we can actually use to try and help us determine where things are. Now, how about animals? Well, the first one that I would normally try is to do a basic transect of something. So for plants, we've got two choices. Plants, we've either got the transect where I'm going from point A to point B, and I'm going to take my quadrat along the entire length, or maybe I'm going to miss so many and go along. We've got the random shape field where you would still divide this up and work out all the locations. So more of random sampling, and we can use those to work out levels of abundance of plants. You can then work out either the frequency of the plant, or we can work out something like the percentage cover. Percentage cover gives us an idea of how much that species is counting up, how much space it's covering in the quadrat. Now, to do that, we've got a more difficult job of trying to measure the area, and this is where the photographs come in really handy, because we can, using certain software, draw round the shapes. We can, having drawn round the shapes, we can then record and measure the area that those objects might actually take up. And that can prove really quite useful when we sort of play around with these quadrats. So we're taking a quadrat, we're taking it across a field, but usually when I try one of these things I do something nasty to my students, because in here we might have the river, and we might do a transect across this, and I always like to make sure if I find a river, we're going to do this, that it's nice to make sure the river is just deep enough that it just goes over the student's wellies. Now, what we need to do with a river is we need to actually look at what we've got. So let's have a look at a sample across a river. And what I've got is I've got a bank here, and I've got some sort of shallow side there, and we look at this and we find that this river, this stream, is going to be possibly deeper one side than the other. And if I want to try and measure this, we often find that using a very simple system such as this type of point quadrat can actually work amazingly well, because what I can do is I can position my quadrat, and how boring with me, I normally take something like a spirit level, and I can put that down above this sort of stream, and then what we do is we drop these needles in, and because they are all the same length, and mine here certainly aren't, we can measure here perhaps the profile of the water. So that's another use for our point quadrat. Having done that, then we can start having a look at what we've got in the water. And this is where my camera can come in really useful. What I would do for this is often I would put down a small quadrat, typically the 50 centimeter one, with its grid, and I put the camera into the water, and with the camera in the water I can then photograph what's going on, so we can see an idea of what we've got. And then what we can do then is start counting up perhaps the individual crustaceans or other life that you are trying to sort of record in the river. And if I'm doing a river, then what we normally do is we start downstream and we move upstream. Now, why is that important? I've got a stream, and this stream is exaggerated, it's going uphill, and if I do some work here at point number 1, I'm going to disturb the water, I may disturb the creatures, I may disturb the mud, and this will flow down. So when I now move to my second position, I haven't managed to disturb any of the life here. I may have disturbed this life, but I haven't disturbed this one. And then if I now disturb this, that's going to move down. If we start at the top and I start mixing it around and disturbing it, then when I come to do this one, I've got quite a lot of the life that I've disturbed in perhaps this sample number 10 in my sample number 11. So we don't want to do that. This one is a much better, and we work going upstream. It's fairly obvious, but it doesn't seem necessarily as obvious to many students when we actually try this and we go out and look at what populations we've got. So these are all basically populations that don't move. You've got plants that don't move, you've got some insects that, and crustaceans and other things that aren't going to move very much in a stream. But how about other animals, and how are we going to try and record those? Well, we've again got various techniques to try and do this, and the first one is capture and release. So what we want to do is simply, we want to perhaps have a trail, and we might put on this trail a trap. Having done this, we then capture what we've got.
Take a Puritan tablets.
We then mark it in some way that preferably doesn't harm.
I'm reminding you. Take a Puritan tablets.
Sorry about that. We then mark the creature. We then release. And then we repeat, so we may here do some recapture, and we can then decide the size of the population. And our population here, which will obviously just be an estimate, so our estimated population will be my total number in the first sample, i.e. the ones that we captured, times the total number in the second sample, the recapture here, over the number of marked individuals in the recapture. And this gives an idea. It gives an idea of the proportion of marked to unmarked individuals in the second sample. Now this does rely on a few assumptions. You're assuming that that population's got a definite boundary, so you're assuming there's going to be no immigration and no emigration. You are going to assume that these marked individuals that you released the first time basically are going to sort of move themselves and distribute themselves around the remainder of the population. You're going to assume that there are few, if any, deaths and births within the population. You're going to assume that your marking, of course, is not going to be toxic and kill the individual, or make it perhaps more conspicuous so it's easier to be eaten. Sort of shoving white Tipp-Ex on them might not be the best way of trying to identify an organism, especially then if it can be picked off by lots of birds. We have to assume that the mark that you've put on there, or the label, actually isn't lost, because that one is also going to cause some problems. And of course, that if you're going to put some sort of trap in there, the organism is going to fall for the same trap a second time. That it isn't going to say, hang on, I recognise that one, I got caught last time, I'm going to avoid it. And that one gives some sort of problem. So mark release systems can work, and we could use this for quite large creatures sometimes. There's an even better way of doing this, of course, and that people forget is to simply use a camera. Now, I've got a camera which is set up with basically infrared, so it can see during the night, and it can capture pictures during the day, so we can get day and night. And it's also got this infrared sensor, so this camera only comes on when it detects either heat, or we can do it with movement. So only when we get either heat, depends what sort of animal you're trying to observe, or when you've got movement, then it's going to record. Now, by doing this system, you've actually got to take a picture of an individual, and since you are not doing this capture, mark, release, recapture system, but you're just observing, you're not doing the capturing, you're not doing the marking, you're not really doing the release, you're just observing and letting it carry on. So you're, if you like, capturing it with the camera and releasing, you're not marking, but you have got a photograph of it. And sometimes the photographs are enough to try and work out this in a population. But there are problems with this, and what I do is I can demonstrate this with a simple experiment. So let's take a field, and inside this field are some cows. So let's have a cow in a field. Okay, now that's easy to spot, easy to work out. But if I want to try and identify how many are in perhaps a wood where I couldn't see them, I can only look at them at a certain point. So it's possible to go along to a field and say, right, now what we're going to do is we're just going to record the number of cows in this field, but we're going to do it by measuring the number of individuals that come through this point. Maybe they don't, so our population might come up with 0, even though there is a population here. Or we're going to find that certain individuals frequent that spot, and perhaps none others do. This is territory, and so I might only find that I get 1 or 2 here, and I get the same number in my second, and that gives me again a false population size. So you are assuming that we've got random distribution. And if I go to a field and we look at an area and we count the number of cows there, they're not as hard to actually work out which ones they are, because they've normally got tags in their ears. And then we do this several times to observe what we've got. We find that the numbers of estimates of the numbers of cows in a field, where we probably do know the exact number, we get wildly different numbers because of the problem with this capture, mark, release, recapture technique. So trying to work out this population is quite difficult. This idea of capturing as many individuals as we can, marking or tagging each individual, releasing them back into the sample area, allowing them time to redistribute themselves throughout the habitat, and then recapture as many individuals as possible and record those, and then trying to use a system to work out the population size. There is a whole series of different ways that we can actually try and do this. There's something called the Lincoln Index. Now, another one we can actually do to try and measure this is to look at not one individual but a whole series of ones, and this is called Simpson's Index of Diversity. And this is a measure of our diversity index, D, and this is going to be concerning N, the number of organisms in the whole ecosystem, and then a small number, N, little n, the number of individuals of each species. And we can say here that D is going to equal the number here times n minus 1 all over the sum of the number of individuals minus 1. Now this gives us a value always between 0 and 1. If you get anything else, you know you've gone wrong. And basically 0 represents no diversity, and 1 is basically an infinite amount of diversity. So the higher the value of Simpson's Index of Diversity, the more diverse the habitat. And we can measure that, and in your exams you'll often be given data that someone has collected, and then you can try and work this out for yourself. And sometimes it's worthwhile trying to work out some of these things. So we've looked at then a few different systems. We've looked at, there we are, Simpson. This one is the Lincoln. Index. I suppose write that down for you. Lincoln Index. And we've got those ways of trying to capture things. Are there any other ways of trying to capture live animals? Well, yeah, it's amazingly good fun when I ask the students to work out how many flying insects, and I give them one of these little butterfly nets, and they try to sort of capture how many insects in an area. Doesn't really work, but it's very entertaining to try and watch students try to capture lots of different flying insects and work out how many in a particular volume. Better is using something like a pooter on something like a tree or collecting some insects. If I say to students, will you collect me some insects, the usual answer is by half of them that no, I don't want to touch them. And so a pooter is a good way of trying to get around this problem. A pooter is simply a container, and it's got a tube in here where you can suck, and we get our insect here, and as you suck in here, so these insects get collected down inside the pooter, and there's usually a little filter on here so that these things don't get sucked back up. So you can collect your insects off perhaps a tree using a pooter. You can use what are properly called sweep nets, and they work quite well for capturing insects, although many students seem to find them particularly difficult. It takes a little bit of effort to try and use some of those so that we can measure the populations. After that, then the only other system is a trap, and we want to make sure that's a humane trap so that you don't actually kill any animals in the process of doing your study. You don't really want to try and interfere with them, and that can mean you don't want to actually sort of stomp all over the place, and that trying to get students to practice on field trips is quite difficult. I hope that's given you some ideas about quadrats. I've done another video on using a quadrat with this camera, and we'll put a link in the description that you can use to sort of see that one in action, because it was too difficult really to try and do this as an outside broadcast live. It's bad enough trying to do it live inside without trying to go outside as well. So if you found this useful, then please subscribe, and I will see you hopefully again on A-level biology, topic by topic, when we'll look at some more biology next time. You take care and stay safe. Bye bye.